So, indeed, as already has been shortly touched upon by Professor Till, extrapolation and switching and or interchangeability are still a few things that are under discussion. Briefly, a few key elements with respect to the concept of extrapolation of indications for biosimilars. What do we mean by extrapolation? This actually means that we will gather data in one clinical indication and we will conclude, we will extrapolate those data to other indications for which we will also approve the product, for instance. And you will see here there are different, let's say, degrees of extrapolation. We can extrapolate from within the same therapeutic area, oncology, for instance, from breast cancer, as has been demonstrated, to gastric cancer, first-line therapy versus second-line therapy, early breast to metastatic breast cancer patients. In other cases, there may also be extrapolation from different disease areas. For instance, in the case of Infliximab, you gather data in rheumatoid arthritis and you extrapolate those data, you will get approval without having clinical studies in, for instance, Crohn's disease, inflammatory bowel diseases. It can even be, these are two autoinflammatory diseases, so you can still say there is a link between those different indications. We will also see, for instance, for Rituximab that is being used in rheumatoid arthritis and in cancer, that one may decide to extrapolate data from obtained in rheumatoid arthritis patients and then extrapolate to cancer patients, which is even a step further. An extrapolation, as I mentioned, means that if we see similar efficacy in one indication, we will also grant approval for use in another indication for which we expect it to also have the same effect as the reference product. This is not just a fancy idea, this is based on the entire concept of biosimilarity, where we actually do not fully rely only on the clinical data, but we actually start with a very stringent and very detailed structural comparison for which we have shown biosimilarity between the biosimilar and the reference product at a level, as you can see here, structural, biologically, in vitro. We have clinical pharmacokinetic and eventually pharmacodynamic data, and one has shown in a carefully, strictly spoken, in a carefully selected study population, the similar efficacy in those particular settings. And when all these requirements are fulfilled, one may decide that indeed these data point towards an efficacy that we can expect in other indications. But you need actually to justify this reasoning. It's not just sufficient to do clinical studies in one indication and then say, okay, we will ask for approval for all the rest. There are quite some requirements. First of all, it's very important to realize that this is only valid, this concept of extrapolation, when the mechanism of action in the one disease for which you have studied your product is the same as the one for which you will extrapolate. And it's obvious that you need to do your first pivotal clinical study in what they then call a sensitive setting. Sensitive in the idea that you should be able to detect possible differences in this particular setting if there would be differences. And that's also the reason why, for instance, in an early breast cancer, this is a more sensitive setting, because then the chances that you will detect differences is higher in this study population. There will always remain some questions, so that's why in many cases you may, after approval, you may have to submit additional data to confirm the justification for this extrapolation. With respect to the mechanism of action, the first requirement that I mentioned, it's important to realize that, or be sure, that we know that the mechanism of action in the different disease states is indeed the same? Is the mechanism of action of Infliximab in rheumatoid arthritis exactly the same as the mechanism of action of Infliximab in inflammatory bowel diseases? There were some discussions or differences in attitude, Europe versus Health Canada, for instance. If we then look at a complex disease as cancer, with multiple cancers that are present, I mean, from a scientific point of view, I think we should be pretty humble and we should not try to state that we know the mechanism of action in detail for a particular molecule in all the different cancers. So there will be always some question marks. And just to give you another overview, it's not only the mechanism of action as such that we need to consider, but also there may be other differences, for instance, differences in pharmacokinetics between one patient population and another patient population that prevents us from this extrapolation, for instance. There may be, depending on the disease state, depending on the type of cancer, for instance, there may be different dosing schemes that prevent us from applying this extrapolation ID. The entire pathophysiology that is associated with the disease may be completely different between the different indications. So these are all things that one has to consider before thinking about extrapolation. Just an example, as I mentioned already, for instance, Infliximab, the pivotal studies were done in rheumatoid arthritis, here in dark blue on the right-hand side, and they were granted approval for all the different indications for which the reference product had been approved. But still, even though not strictly required by the European authorities, the company did also include studies in inflammatory bowel disease to convince physicians that indeed this extrapolation issue was warranted. For Rituximab, for instance, a very recent biosimilar that have been approved, you may know that it is being used in different types of cancer, here indicated as MabThera on the one hand, and it has approval in the different types of cancer and in rheumatoid arthritis. With respect to the first biosimilar for Rituximab that was approved, there were also only done clinical studies in rheumatoid arthritis. They were granted approval for the different indications. In the meantime, during the approval process, this is not finished yet, there is also a study that has been set up in a cancer, in a lymphoma setting. But these studies are still going on. So the approval is based, the extrapolation is based merely on the rheumatoid arthritis studies and then the extrapolation to cancer. If you look at another Rituximab from another company, they did the reverse. They did a pivotal study in lymphoma patients, and they got approved for all the indications, and they are currently running a study in rheumatoid arthritis. So this also illustrates that the one biosimilar company may have another approach than the other biosimilar company. So you cannot conclude anything by looking at one biosimilar file. You will have to look in a little bit more detail. Now, another important issue, interchangeability, very briefly. And when we discuss interchangeability with respect to biosimilars, we need to, or I want to remind to you again that the discussion starts from the concept of biosimilarity and from the fact that we know that biosimilars are not identical to each other and not identical to the reference product. That's the starting point that we have to keep in mind. Interchangeability, what do we mean by interchangeability? Interchangeability is not an opinion. It's not a matter, do I feel that the two products are interchangeable? No, actually the interchangeability is a scientific property of two drugs for which we have demonstrated or for which we know that changing one drug for the other drug is assumed to result in the same clinical effect in a given setting to a patient. And when we talk about biologicals that are being administered more than once, we should also realize that when we put the label interchangeability onto those products, that it also implies that alternating, going back and forward between the two drugs does not have any impact on the safety nor on the efficacy in those patients. And that's why also, I won't buy the license. Yep. This also immediately brings me to the point that when introducing biosims, we should make a distinction between treatment-naive, bio-naive patients on the one hand, and patients that are already under treatment. What does the regulatory agency say? Europe doesn't say anything. They don't, if I can say so, they don't take their responsibility. They just say it's a matter of the national competent authority. So each country in Europe decides for themselves. But as I mentioned, it's a property of a drug, so it's not a matter of deciding by individual competent authorities to me. In the US, they take the mere scientific approach, and they make a clear distinction between biosimilarity and between interchangeability. Interchangeability is a step higher, a standard higher than biosimilarity. So once you have demonstrated biosimilarity, you may decide to try to demonstrate interchangeability. So to demonstrate that if the product is administered back and forward, those two products back and forward to patients, that does not have any impact on the efficacy and so on. There are draft guidelines, they are not finalized yet as of today, and obviously there is no biosimilar so far that has also been submitted or that has been approved as an interchangeable product. You have seen a few possible studies also in the previous presentation. Just very briefly, how will we demonstrate, or how should you demonstrate biosimilarity? There are quite a number of different studies that you could design, for which the one at the bottom, the multiple switch, is the most relevant one, I believe, which is still not very simple, because how long should the treatments be? The blue one, the orange one that is indicated here, should this be 6 months, 1 year, and so on? What do you need to follow up, and so on? It's clear that the design, the bottom design, the multiple switch, is probably from a financial point of view not feasible. So in the best case, we will have to do it with a single switch crossover or with a single switch parallel arm. For the sake of time, I will skip this one, but you will understand that when you switch a patient, first of all, there are quite a number of scientific questions that can be raised with respect, for instance, the long half-life of a monoclonal antibody. So you will always have a situation where the two products are circulating in the body of the patients. There's also the ethical considerations that you have to make if you have a patient that it took many years to get the right treatment. Now the patient is stable. Will you then take the possible risk, even if it's very small, will you take the risk to switch the patients? And if I say switch, it goes in both directions. If a patient is stable on the biosimilar, then the same concern holds. I mean, you should not switch to the reference product, and vice versa. There are only few studies. Whatever they may try to tell you, there is no solid scientific information yet on switch data. But there is the Northwich data and there is the DAN Bio study. I will just focus on the Northwich study, which is a study which was sponsored by the government, approximately 500 patients for Infliximab, one single switch. It's important to realize that those 500 patients is a very heterogeneous population of patients with, on the one hand, inflammatory bowel disease, on the other hand, with inflammatory joint diseases, and also with plaque psoriasis. And within those different indications, also different sub-indications, rheumatoid arthritis, spondyloarthritis, psoriatic arthritis. So this is a very heterogeneous population, only 500 patients. It was a single switch, parallel arm study, and it took, the duration was 1 year. Now, without going into too much detail, these data have been published in The Lancet in early this year. And these are the conclusions. You can see here the non-inferiority margin was set at 15%, and it was fortunately concluded that the biosimilar was not inferior. This just illustrates that indeed under those conditions it is confirmed that this is a biosimilar, but this does not conclude anything about switching, obviously. It is also concluded that the study actually was not powered enough to show non-inferiority in individual diseases. I mentioned to you that this was a very heterogeneous study population, so we cannot tell anything about, for instance, Crohn's disease, because the subset of this patient population was much too small. And it's definitely such that all those data do only tell us at best something about Infliximab, about one biosimilar of Infliximab. It does not allow us to generalize to other Infliximab biosimilars, nor to biosimilars in general. So when it's clear that also in this study, even though it was for one biosimilar Infliximab, it only holds for one single switch in one direction and does not tell us anything about possible multiple switches. So that's why the authors clearly state that we will need more data. And even if we have data, even if you spend quite a lot of money to demonstrate clinically that switching is indeed fully safe and does not have an impact on the clinical efficacy, we also have to take into account that biosimilarity is only valid at the time of approval. And this has also important consequences for interchangeability. We know when a product has been approved, a biosimilar, it was biosimilar at the time of approval. We should realize that there is no guideline to double check, to reconfirm biosimilarity after a manufacturing change of the originator or of the biosimilar. This actually means that it is possible that for particular critical quality attributes, the two products shift from each other in opposite directions. So even though the intrinsic quality remains okay, even though both products remain safe and effective in a patient, it could happen that after 10 years, the biosimilar that we have approved is not, from a statistical and scientific point of view, not similar anymore to the reference product. So that's something we have to into account. So this also means that if you have proven interchangeability at a certain time point, you have no idea whether this conclusion on interchangeability remains valid after 5 or after 10 years. So it's something, this is a remaining question mark. Another very important point when working in real life is the fact that you know that for quite a number of antibodies and other products, we have different independently developed biosimilars onto the market. Biosimilar 1 has not to be compared, has not to demonstrate that it's biosimilar to biosimilar 2, for instance. This is completely different compared to the generic situation where overall, if you have a generic that is generic A that is identical to the reference product, and you have an independently developed generic B that is identical to the reference product, because of the identity, you may conclude that in 99.9% of the cases, generic A will be exactly the same or identical to generic B. This is completely a different situation with biosimilars, because biosimilars are similar to each other, to the reference product. So if we consider a biosimilar A for which we have demonstrated based on statistical information that the differences are not statistically different between the properties of biosimilar A and the reference product, that's okay. If we consider an independently developed biosimilar B for which also we have demonstrated by statistics that the differences that we observe are not significantly different between biosimilar B and the reference product, then I invite you to go back to your course of statistics to know whether or not you may conclude anything about the statistical significance of possible differences between biosimilar A and B. And there's no way you can make any conclusion between the possible difference of A versus B. This also has obviously its impact on interchangeability. There's definitely no way to believe that you would have any data on possible interchangeability between biosimilar A and biosimilar B. And that's where we possibly include the risk if somebody says biosimilars are interchangeable with the reference product. This also means that on the floor, people will think that you can also interchange biosimilar A with biosimilar B, which is definitely not proven by any means. And this will indeed be a possible situation in real life, in hospital, but also in the retail setting on one day. So consider the situation where we have one biosimilar, then it's pretty simple. We only need to look at what the patient has been giving, the green biosimilar or the green reference product, or the orange biosimilar B1. But for Infliximab, for Rituximab, we have at least 2 biosimilars onto the market right now. This means that when the patient has been treated with the originator molecule, and then maybe 6 months later with the biosimilar B1, it could happen after 12 months that maybe the tender goes to the blue, to biosimilar B2, and then the patient may be subjected to biosimilar B2. And for some biosimilars, we have much more onto the market. So this means that actually you may end up in a situation where it's very difficult to, even if you would consider, but I would advise not to consider, but if you would consider switching the patients, that it may be difficult after 1 or 2 or 3 years, that it may be difficult to trace back which version has been given to which patients at which time point. And if you observe an adverse event, it will be very difficult to trace back which product may have been responsible or not been responsible, because one of the major adverse events that we are concerned about for biologicals is immunogenicity. Immunogenicity takes some time to occur. So if 6 months after the switch you see an immune response and you have already 2 switches, then it's very difficult to unambiguously define which product was responsible for the immune response. And maybe it was not the particular product, but maybe it was a switch. So also in terms of pharmacovigilance, traceability and so on, it's strongly discouraged, I would say, to consider switching on a basis. So to conclude, there are many abstracts with studies, small studies, single switches, 10 patients, 20 patients, that have demonstrated no big issue with switching. But you will realize that these studies are not statistically powered to make any conclusion. So with 10 patients you cannot detect any possible issue. So there are no evidence-based, clear, solid data to support multiple switching between biosimilars and the biosimilar and the reference product. If you would start with, this also implies that actually substitution should actually not be allowed, because it will create problems with safety on the one hand, but also it will compromise the traceability of the product. And it's definitely not to be overseen anymore in a decent way when we have multiple biosimilars onto the market. And even if you have demonstrated interchangeability, this interchangeability label will have an expiry data because of the possible manufacturing changes that may have occurred. So that's why I believe that no problem with any good quality approved biosimilar, but we should restrict its use to treatment-naive patients to have a safe implementation of biosimilars and also a safe attitude towards the patients in general. And with that, I will finish.